Thompson. I'm an assistant professor in biology. Picocyanobacteria are a type of bacteria. They're very small and they do what's called photosynthesis. In that role, with that process, they do what plants do on Earth, but they do it in aquatic systems. And so they form the foundation of food webs in the ocean, in lakes and rivers. But they also make oxygen, which is really important to our lives and shapes the life of everything on the planet. So our project on the Great Lakes looks at Picocyanobacteria, so very small cyanobacteria that live in the Great Lakes. And I'm working with Maureen Coleman and Jake Waldbauer, who are at the University of Chicago. And several years ago, they started a time series to sample across all the different Great Lakes. And going into the project, they knew that there were small cyanobacteria there, but they're kind of all lumped together as one group. And I developed a new method using a technique called flow cytometry to be able to actually pull apart and see these different subtypes of Picocyanobacteria. So using that method, we discovered about 10 subpopulations or 10 different types of small cyanobacteria that live in the Great Lakes. And we're showing that they have different distributions across the lake. Our work is showing that there are distinct environments within the lakes. And this is really important because so many people depend on the Great Lakes for economies, for good water quality for their lives and recreation. And really what we want to do is be able to quantify or count each of the different groups that we find. Picocyanobacteria are present in all aquatic systems that get hit by sunlight. And so they're in lakes and rivers, but they're also in the ocean. And one of the things that's special about the open ocean, so really far from land, is that almost all the photosynthetic cells there are Picocyanobacteria. So we try to understand how these cells get through their day, how they divide, how they grow. And one thing we know about these cells is that they're eaten by other cells. We see that about half of the population of cells dies every day, or I should say it's lost every day. And we have an idea of a few things that are eating these Picocyanobacteria, and part of this what's called a microbial loop. So this is how microbes use carbon and transfer that carbon to larger animals. But there's a huge chunk of this loss that we don't know why it's happening. And that's where our work comes in. So we're interested in a group of grazers that are called pelagic tunicates. So these are tunicates that live in the surface ocean, so they swim freely in the surface ocean. And on my most recent field trip, we focused on a group called salps. We've looked at several different types of salps. They come in all kinds of different shapes and sizes. And we think that these differences in the salps could be relevant to how they filter bacteria, especially our Picocyanobacteria, from the ocean. This project has a few different objectives. One of our objectives is to basically document whether these salps or gelatinous grazers are actually eating bacteria. Are they eating the smallest cells in the ocean? The Picocyanobacteria are really important to the carbon cycle. They're important to making oxygen. And so these are things that ecosystem modelers want to be able to capture and quantify in a model so that we can say, in 100 years, under this climate scenario, how many of these cells will be there and what will they be doing to the carbon cycle. My name is Ann Thompson. As a microbial ecologist, I study how very tiny cells, through their activities, can impact the entire Earth system and shape life on Earth as we know it.